All right, here we are looking at various things that we can see from the peak now that we have a basic knowledge of molecular biology, as it's now called. I kind of got maybe wandered a little afar on looking at sports when we talked about mutations, because that's something people can relate to. But just to tie in Wednesdays and Mondays lectures, in case this wasn't clear, the reason why we keep seeing new strains of virus is mutation. Virus DNA or RNA, like any nucleic acid, is subject to copying errors. Whenever you copy it, there is a non-zero chance that one of the nucleotides is going to get changed, or possibly a section will get deleted or sometimes duplicated. Sometimes even stranger things can happen. The majority of these errors either don't have any effect at all or they've got a negative effect, but there's always a few that may enhance a virus's, or for that matter, bacterium's ability to spread, or its ability to dodge an infected person's immune defenses. And this, by the way, is why you have to get a flu shot every year. There's not one single flu virus. The flu viruses are constantly mutating, and mutation constantly generates new strains of the influenza virus. And groups like the Center for Disease Control try to get out ahead of the parade and predict which new varieties of flu virus are likeliest to be a problem, and create a vaccine for those and give it to people before flu season hits. And there are times when their estimates are incorrect and the vaccine's not very effective. So you can get the flu after having your flu shot. Fortunately, how to do this is well understood, and they've got the practice, they've got that procedure down to a routine. But still, every so often you can get hit with a strain of flu that you weren't vaccinated for, because flu is constantly coming up with new strains, new varieties that can dodge your existing immune defenses. This is also part of the reason why it's been very difficult developing a working vaccine for HIV. HIV has an unusually high mutation rate, and even within one patient's body, HIV viruses are constantly mutating. You're always getting new varieties of HIV, most of them either neutral or not as good as the ones you're infected with. But every so often you'll get one that's better at infecting, at staying in the body, at dodging the body's defenses. And HIV eventually overwhelms the body because it mutates faster than the body can develop defenses against it. And that's one of the factors that's made it very hard to develop a vaccine for it. You might have been hearing in the news about a variant of COVID-19 called B117. In the center right here, you've got a little diagram of what the actual virus looks like. That's the virus. You might remember that on its outside, it has what are called spike proteins. That's these things that look like sun rays or points on a crown. And then right here, you've got the nucleic acid inside all of this. Surrounding all of that, you've got a diagram. This colored curve is a diagram of the virus's RNA, where different colored parts code for different proteins. And this red zone right here, that is a section of the virus's DNA that codes for those spike proteins. Compared to the original strain that we found, the spike proteins of B117 have 6 point mutations, places where one amino acid has been replaced with a different one compared to the first COVID that was detected. And then 2 points right here where there's been a deletion, where an amino acid has actually dropped out of the finished product because some bases have been cut out and lost. That's actually what makes B117 more infectious and why there's some risk, although it doesn't seem to be a major problem yet. But there's concern that a variant like B117 could bypass vaccine protection. This is a computer model of that spike protein on COVID. And you might recall that the spike protein is what the COVID virus uses to stick to a protein on the membrane of your cells. Right? This is how the COVID virus anchors itself to your cells before getting pulled inside. It uses the spike protein. B117 has one mutation in particular, one of those 6, that changes this amino acid right there, amino acid number 501, from the amino acid asparagine to the amino acid tyrosine. You wouldn't think that's a big deal. I mean, there's over 1,000 amino acids here, so this is like changing one of the screws in your car. You wouldn't think it would have a big effect on the whole thing. But that one change seems to improve the virus's ability to stick to those cell surface proteins. The original COVID would get into your body, and the spike proteins would stick to proteins on the outside of your cells, but they didn't fit really well, and they'd often fall off. Kind of like the ball hitch I was towing on a trailer a couple of days ago that also kept falling off whenever I did something crazy like exceed 20 miles an hour. Long story there that I'm sure you don't want to hear. But original COVID would fairly often slide off. This one sticks to cells better, which means that it's more infectious. There's also concern. Remember that if you've had any of the vaccines, they make your body make antibodies to the spike proteins. If you change the shape of the spike proteins even a little bit, your antibodies may not work as well. So that's the reason why, people are worried about variants like this. And the reason they're here is mutation. And the reason they spread is plain old natural selection. Good old Charles Darwin does it again. You might remember that natural selection is based on the principle that individuals vary, and that variation can be passed on, and some of that variation improves the odds of surviving and reproducing. Some of those mutations in a virus improve the virus's chances of passing itself on. So natural selection is the reason why some of these mutants not only appear but spread. Might almost be something into that stuff that old Darwin guy used to say. Now, I actually went online about an hour ago and was able to download the complete gene for the spike protein, and this is it in a database called GenBank, hosted by the US government on a site called the National Center for Biotechnology Information. With a little familiarity, you could do this too. You could download the complete COVID genome, all of the genes that it's got. And the spike protein is coded for by a gene that goes ATG, TTT, GTT, TTT, CTT, GTT, TT, ATTG, CCA. Hope you get this memorized by the time of the exam. Actually, I'm kidding. But you might be wondering how we can read virus DNA or any DNA like this. How do we know that this is what the COVID spike protein gene says? How can we read those nucleotides one after the other? Well, to find out, We have to look at the other great scientist you know who got his PhD from the University of California at Berkeley. There's actually two. I'm one of them. The other was a guy named Kary Mullis, shown here. And back in the 80s, he was working for a biotechnology corporation called Cetus. I'm not sure what happened to Cetus. I'm reasonably sure they got bought out and bought up and changed their name, and they're now a division of, oh heck, I don't even know. But he was working for this corporation back in the 80s and surfing a lot in his off hours and taking a lot of drugs in his off hours. And in 1955, after taking a sizable dose of LSD, he was inspired to come up with a technique that would take any piece of DNA and copy it exactly millions of times over. You could take even the slightest little snippet of DNA and copy it and copy it and copy it over and over and over again. To be fair, there were other people who'd come up with similar ideas, and Mullis was not doing this on his own. You know, some people think of scientists as being, you know, kind of like, you know, lone wolves in their laboratories, you know, staying up all night, you know, completely isolated from the world as they strain to unlock the secrets of the universe. But the fact is, a heck of a lot of science, probably most of it these days, is done in teams. And Mullis was part of a team, you know, not a lone, you know, mad scientist up in the castle or something like that. But he was the prime mover in making his idea work, and he won the Nobel Prize in Chemistry in 1993. He's probably the Nobel Prize winner in the past 30 years who looks the best with his shirt off. And he wrote this book called Dancing Naked in the Minefield, which reads about like how it sounds, reads like the autobiography of a brilliant scientist who's taken a lot of LSD. I should mention, by the way, that way back at the beginning, I talked about coming up with hypotheses. And I mentioned that coming up with a good hypothesis was, in fact, a creative action. I certainly am not recommending that anybody try drugs as an avenue to get more creative. John Lennon took a lot of drugs, but claimed that they didn't make him write anything that he couldn't have written without them. But it is true that hypotheses can come from very strange places. And in this and at least one other case I can think of, scientists have actually come up with hypotheses while tripping. The difference is that they sit down and test them while they're sober, instead of just going, oh, wow, dude, DNA, dude, it's fuchsia, whoa, right. What Mullis came up with is called the polymerase chain reaction. For short, it's called PCR, involving a lot of these little mini plastic test tubes that I think you should have worked with in, did we do this in one of the hands-on labs? Did you do the hands-on DNA fingerprinting lab? I think you did. I hope you did. And if we did, you probably saw tubes like that. And PCR makes use of a DNA replicating enzyme. Remember that enzymes are what do the actual work of replicating and transcribing DNA. In particular, this one is called DNA polymerase, harvested from a bacterium called Thermus aquaticus, which we often abbreviate as just TAQ, TAC. So TAC polymerase. And Thermus aquaticus happens to live at unusually high temperatures. It is, in fact, an extremophile, which is a word you ought to remember from about week 3 when we were talking about archaeans and the way that many of them live in environments that are too hot or cold or salty or acid or alkaline for anything else to live. That's an extremophile. Thermus aquaticus is an extremophile, and that's important because its proteins are adapted so that they don't denature, right? They don't change their shape and lose their function when the temperature is hot. So to do this, you just heat your DNA, and if you heat DNA, you will cause it to unzip. You will unzip your genes, not very funny, breaking those hydrogen bonds that lock the strands together. Then you hit it with this. This is the DNA polymerase. This is a computer model of it. Lots of alpha helices there. That DNA polymerase, since it comes from an organism that is naturally able to live in high temperatures, about 60 Celsius, that's what, roughly 150 Fahrenheit, the DNA polymerase will go to town. It can work on its own outside of the cell that it came from, and it will build new strands using those old strands as templates. And then you cool it down, and then you heat it again and repeat this over and over, cycling it through heating and cooling. And every time you heat it, the strands unzip and the Taq polymerase builds new strands. And then when you cool it, the strands come back together. And then when you heat it again, the strands unzip again, and you keep doing this until you have millions of DNA pieces. There's a little bit more to it than that, but that gives you the basic idea. Sorry, just checking this something on the chat, see if I need to take care of. There's a nifty little way you can modify the procedure that I don't have time to get into, but it's pretty simple. And this makes it possible to directly sequence DNA. So you can take a single piece of DNA, copy it millions of times over, and then do a technique called gel electrophoresis, which is what you should have done in that DNA fingerprint lab that hopefully you did. And you can read off the order of nucleotides. And we'd read them here from left to right, and each lane corresponds to a particular nucleotide. So we'd read this from left to right, and we'd read GG, GG, CTTAG, sorry, GGC, TTC, GAA, GGGGAC, TACAAA, GGGG. It actually kind of reminds me of musical notation. You could actually sing your DNA if you wanted to. You could go, let's see, G G C T T C G A A G G G G A C T A A C A A A G G G G A G A G C T. I'll stop there. It's not a very interesting tune, but there are people that have actually experimented with trying to compose music based on DNA, based on assigning pitches to different nucleotides. So that's how you read a piece of DNA with a modified version of PCR. And that's what makes it possible to identify DNA samples from individual people, even if you only have the tiniest amount of DNA. You can have, just a little bit of saliva, cells from a hair follicle, a small drop of blood, sometimes a semen sample. Sorry, but it happens. And you can take that, copy the DNA many millions of times over, and sequence it and read anybody's DNA that you want to. And this is what makes it possible to identify DNA samples. In particular, we don't sequence, your entire DNA, when we take a sample like this, we use a set of techniques called DNA fingerprinting. There's more than one way to do this, and I can't talk about all of them in the time that we've got. But the most common method takes advantage of a fact that not all of the DNA that makes up your chromosomes actually codes for proteins. There's parts of it that are involved in controlling transcription and translation. There are parts of it that we're not sure what they do. There are parts that do nothing. And there are parts of your chromosomes that are called STRs, standing for a short tandem repeat. And in a short tandem repeat, the nucleotides don't make up a gene. They're usually not transcribed and translated. You just have a short sequence of between 2 and 6 nucleotides that repeats over and over. So somewhere in your DNA, you might just have AT, 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 or CATG, 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 right? That CATG repeated over and over. Mostly this is thought to be just junk DNA. It doesn't seem to do anything. And one of the things about STRs is they can vary in length. There could be some piece of one of my chromosomes where I have CATG repeated 7 times, and one of you might have it in the same place, but it's repeated 9 times. And somebody else might have it 13 times, and someone else might have it 8. And this makes no difference. You know, there's no function to these STRs. There's no way to tell that I might have 5 STRs and you might have 12 without going and getting our DNA sequenced. And usually there's no reason for that. But STRs vary in length, and we can use PCR and gel electrophoresis to measure the length of certain DNA fragments. So I could take a blood smear from a crime scene, copy the DNA millions of times over, isolate the STRs, and measure how long they are. So in case anybody's forgotten, in gel electrophoresis, you put DNA samples into little rectangular wells in a slab of soft material made of a carbohydrate called agarose. That's the gel. Once you've loaded those little holes with DNA samples, you apply an electrical current to the gel. And since DNA has a slight electrical charge, it will get pulled towards one end of the gel by the electric field. And so small fragments of DNA pass through the gel more quickly. So you end up sorting DNA fragments by their size. Okay, hopefully everybody remembers how this was done. I think you all did this in lab. So it's pretty straightforward to sort pieces of DNA by size. And so we could find a piece of one of your chromosomes that's an STR, copy that piece, and then count how many repeats there are. Now, for any single STR, somewhere between 5 and 20% of the human population will have the same length. But there are tens of thousands of STRs, tens of thousands of places in your chromosome where instead of having a protein coding gene, you have a repeated CGA, 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 or something like that. You know, it would be a little bit like reading a book, and on every page, there's a place where, you know, the prose comes to an end, and you read, "In a hole in the ground there lived a hobbit, blurp, 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 blurp." Not just any hole, right? And different copies of the book had different numbers of blurps. Now, there's tens of thousands of STRs. What the FBI in the United States does through a computer system called CODIS they store the length of 13 core STRs, and more recently, they've started storing 7 more. So if your DNA is in the FBI's system, what that means is that they have not your entire gene sequence, not all of your genes. All they have is the lengths of your DNA sequences at 13 or now 20 locations on your chromosome. They just store some numbers. And the probability of 2 people that are not like identical twins having all 13 STRs match up is 1 in a quintillion. There is 1 chance in 1 followed by 15 zeros that 2 people who are not relatives should have identical STRs at all 13 locations. And this happens to be a map of where those 13 are. Each of these bar diagrams with the stripes is a diagram of a human chromosome. And each of these yellow boxes with numbers in them is the location on the chromosome of one of those 13 STRs. So there is a location on chromosome 2 that's called TPOX, which I always thought was the guy who had the big beef with Biggie, but I guess not. TPOX is just a name for a location on chromosome 2 that has one of these repeats. And there is a location on your chromosome 3 and mine called D3S1358. And at that spot, there is some sequence that repeats over and over again, blurp, 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 blurp. And different people can have different numbers of blurps. And so on. All of these little yellow boxes with code numbers mark the locations of the STRs that the FBI takes an interest in. And this is a part of somebody's DNA fingerprint. You certainly don't have to memorize this, but right up here, this says D19S433. That's the code for one of these STRs. There's a location on chromosome 19, and this person has 2 copies of chromosome 19, one from his mom and one from his dad, and one of his chromosome 19s has 13 repeats at this location, and the other has 14 repeats. And then there's a location on chromosome 12 that's called VWA, and this person has 2 copies of chromosome 12. One of them has 14 repeats at VWA, one has 17. There's TPOX right there on this guy's chromosome 2. He's got 2 copies of chromosome 2, and both of them happen to have 11 repeats, and so on. So this is the data that's stored in a genetic fingerprint. They don't have to sequence all of your DNA. Now, when the technology became available, people were really worried about privacy issues. You normally have to, the cops normally have to get a warrant and or get your consent in order to search your house or your vehicle, but you shed DNA every day. It's in your dandruff and your sweat and your saliva. And there was concern that the police would be able to do all kinds of intrusive evidence gathering without you even knowing about it. And there certainly are ways that DNA can be misused. I'm not going to say this is a neutral technology. On the brighter side, though, DNA evidence can be used to overturn wrongful convictions. I just checked their website, the Innocence Project, a couple of days ago, and as of this month, 375 prisoners over the past 20 years or so have been exonerated using DNA testing and released from prison. This includes 21 who were on death row. The gentleman that you see here in the center of the picture, Levon Brooks and Kennedy Brewer, were imprisoned in Mississippi for raping and murdering children. But DNA evidence showed that they could not have committed the crime. And after entirely too long, they were both freed in 2008, thanks to DNA testing and the work and the fundraising of this group called the Innocence Project, that helps with legal representation as well as scientific help. Oh, I don't normally make pitches like this, but if you're concerned about injustice in this country and would like to do something about it other than wringing your hands, the Innocence Project is one place where donations can actually make a real difference in uh, human lives and can bring, albeit delayed justice, can bring justice to unjustly accused people. So, yeah, it would be, something good for you to investigate if you're thinking about making charitable donations. Right. Different groups of people tend to have different sets of STRs. Now, STRs are minor in that they don't affect your gene function. None of them are better or worse than any others. They're just differences between people. And they don't necessarily correlate with what we think of as races. Race is not really a biological concept at all. People vary, but that variation doesn't neatly pigeonhole into what we think of as human races. That being said, we can sometimes use these to trace human ancestry. Men on the Y chromosome have some of these STRs and some other variations, and one set of these variations happens to be called haplogroup Q. And what's shown here is they've taken DNA samples from people around the world, and you can see that haplogroup Q, this set of STRs and other DNA variations, happens to be very common among the native peoples of North and South America. It also happens to be common among native peoples of Siberia up here and a region in Siberia right there. Now, you might have heard that Native Americans are thought to have migrated from Asia this way across this little strip right here called the Bering Straits, which is thought to have been dry land about 13,000 years ago. Well, now we have some genetic evidence for that there are some gene sequences. They don't really do anything, but they're shared by North and South American natives and certain populations in Siberia. So those populations are probably related to each other, which is what we would expect. If the ancestors of Native Americans traveled this way, they probably had relatives that stayed behind. And well, this seems to be where they are. If you get your DNA tested by 23andMe or any of those companies out there, this is what they test you for. This is the kind of thing that they do because there are other STRs that tend to be more common in people from other parts of the world. And so you can sometimes tell where your ancestors came from by trying to match up STRs. Like this is basically what's in those. This is the data behind those gene testing kits that you can buy. Last thing I'll talk about is that if you can sequence DNA, you can not only understand human history, we have whole new lines of evidence for reconstructing evolutionary history. I actually went into the GenBank database and was able to download short snippets of DNA from a hippo up at the top and 3 different whales. And if you line them up, you'll notice that at some points, the nucleotides are identical. So like right here, all 4 have a T right here. And then for this nucleotide, the hippo and the orca have a G, but sperm and blue whales have an A. And then all four share a C, and all four share another C. And then the hippo's got a C, but the other three have A's. And then hippo and blue whale have G's, but orca and sperm have A's, and so on. And so you can see there's differences in individual nucleotides. And here's the thing, if two species share the same nucleotides, the simplest explanation is that they descend from a common ancestor. And these nucleotide differences result from mutations in the past. So we can take sequences like this, use computers to find all of the differences, nucleotide by nucleotide, weigh up the similarities and the differences in DNA using various means that we don't have time to get into. This is kind of a big field. And we can draw cladograms, which you might remember from about week 5 as diagrams of hypotheses of common ancestry. And this is a recent one. All of these little markers right here are different kinds of genetic differences that are shared by these animals over here. And you might have noticed that here's whales. Toothed whales includes orcas and sperm whales and also dolphins. And these share a common ancestor because they have these genetic similarities, but they also share similarities with hippopotamuses, which seem to be the closest relatives of whales that are not whales themselves. Whales are descended from land mammals. And in fact, they're descended from a critter that was also ancestral to hippos. And then the next closest relatives to whales and hippos are pecorans. That's giraffes and deer. It's ruminants, so it's also cows and goats and sheep and antelope and things like that. And then their next closest relative after that is pigs and peccaries. And then beyond that, their most distant relative on this cladogram is camels. So whales actually come from within this grouping of hoofed mammals. And we have fossil evidence that backs that up more than we can talk about, but it's confirmed by similarities in DNA that whales share with hippos, but nobody else. So that's the fun stuff you can do once you're able to sequence DNA. We can use it to explore the evolution of all life on Earth. That's a cladogram of certainly not all life, but it's a cladogram of all of the species whose DNA we have significant sequences of as of a couple of years ago. And yeah, believe it or not, we're right about there. We're in a grouping of animals. On this slide, they're called metazoa. Our closest relative to the animals seems to be right here, and it's the fungi. These are various groups of protists. These are some more protists in here. The plants come out here. That group called Archaeplastida includes plants and some other protists. And then the archaeans are down here, the bacteria down here. Even the bacteria and the archaea have deep genetic similarities with you and other advanced animals. You share something like half of your DNA in common with a banana, which is over here in this group called the Archaeplastida, which is what includes the plants. And to end us where we began, we can use the same approach to track the evolution of virus strains. This is another cladogram. These words and letters right here are just names for different viruses that have been isolated, some of them from humans, some of them from other people. This one right here called human MERS-CoV was the causative agent for it, was called Middle Eastern Respiratory Syndrome. which could have caused a pandemic but didn't really go global. But that was a health concern several years ago. And then COVID is all down here, and its closest relatives are viruses that are found in bats. And so we can track the evolution of virus strains. We can work out where viruses come from. We can watch as new virus strains evolve and use that to try to defend against them. So we can track all of this mutation plus natural selection and then use that to develop better vaccines and better defenses against them using DNA sequencing as one of our tools. So hopefully what I've been able to do is tie in a lot of very different material, different themes by talking about this. And you see I've made some callbacks to material from earlier in the course. I do that a lot. And this is why you need to know everything and why the exam is going to be cumulative, because it all ties together. All right, I will now